morning, everyone. I'm Ava Kadiwada, an internal medicine chief resident. Welcome to Grand Rounds this morning, where we have the Longer Life Foundation. We'll get started this morning with a few words from Dr. Dan Zimmerman, the managing director of the Longer Life Foundation.
Good morning, and welcome to Internal Medicine Grand Rounds. As noted by Dr. Kadiwada, I am Dr. Dan Zimmerman, and I lead the Longer Life Foundation, which is a collaboration between Washington University and Reinsurance Group of America, based right here in Chesterfield. Now in its 23rd year, the Longer Life Foundation's mission is to fund and support the study of factors that either predict the mortality and morbidity of select populations or influence improvements in longevity, health, and wellness, such as the research we funded by Dr. Grant Challen, our speaker today. Since 1998, we have funded over 130 research grants here at WashU. As of last week, our call for letters of intent for this year just closed, and we received 19 new proposals. So we are definitely looking forward to reviewing those in the coming weeks. With that, I would like to turn things over to Dr. Dominic Reeds, Associate Professor of Medicine and Medical Director, Nutrition Support Service at Barnes Jewish Hospital. Dr. Reeds also serves as Director of the Longer Life Foundation's Longer Life Center here on the WashU campus. Thank you.
Thanks, Dan. Good morning, everyone. Thanks for joining us for Grand Rounds this morning. We're extremely lucky to have Grant Challen speaking this morning. Grant is an incredibly successful researcher in the field of hematology oncology. He did his BSc with honors in molecular biology and a PhD in developmental biology in Queensland, Australia, I believe it's pronounced. He did a postdoc fellowship at Baylor College of Medicine. We were lucky enough to recruit him to come here in 2012. Grant has really just excelled coming here, is extremely well funded and runs a really strong and cohesive and very interesting research program into the pathogenesis of a variety of hematologic malignancies. And without further ado, thanks very much, Dr. Challen.
Great. Thanks very much, Dr. Reeds and Dr. Zimmerman. And it's a pleasure to be here today presenting on behalf of the Longer Life Foundation. So today we'll cover a bit of our lab research, understanding clonal hematopoiesis, including its regulation, mechanisms, and potential opportunities we have for blood cancer prevention. So aging is the #1 risk factor for the development of cancer, and the blood system is no different. Age-related changes in the hematopoietic cell compartment over time underlie many of the pathophysiological deficiencies seen in the elderly. Now, the vast majority of these stem from the functional decline of hematopoietic stem cells with age. This leads to decreased immune cell function, making this elderly population particularly sensitive to COVID and other infections. And this correlates also with an increased risk of myeloid malignancies over age. And really, the central mission of our laboratory research is to develop ways to extend hematopoietic health span and reduce individuals' lifetime risk for blood cancer development. So really, the foundational building block through which we like to view these pathologies is that of the long-term hematopoietic stem cell. So these are the very unique and special cells that live in your bone marrow and regenerate your blood system for as long as you live. They have two distinct properties which separate them from the rest of the blood cells. That is the ability for long-term self-renewal to make more copies of themselves and sustain this population for as long as you live. And the capacity for multi-lineage differentiation to generate all of the mature effector cell types found in your blood and bone marrow. And although hematologists like to draw this stepwise hierarchy in the development of the blood system, which we've used for convenience over the last 4 or 5 decades, the advent of single cell technologies has really shown us that this differentiation process is really more of a continuum rather than these discrete cell subsets. Although the classical model does prove useful for us when thinking about developmental fate decisions and isolating cell populations for functional interrogation. So over time, hematopoietic stem cells will randomly acquire genetic mutations as a consequence of aging. They average about 1 to 2 mutations with every cell division. Now, 99% of the time, these mutations are inconsequential. They happen in non-coding regions of the genome or genes for which stem cells simply don't care about. But very, very rarely, these mutations will happen in genes which are crucial for hematopoietic stem cell function. And if this mutation somehow increases the relative fitness of that cell, then this clone with bearing this mutation will expand and gain clonal dominance in the bone marrow over time. So this phenomenon happens in basically everybody as we age, and it's been a condition that's termed clonal hematopoiesis, the detection of age-related mutational signatures in the blood of otherwise healthy individuals. Now, while most of us will develop clonal hematopoiesis at some level over age and live normal, healthy lives, it is actually a risk factor for the development of subsequent myeloid neoplasms, as well as a range of other non-hematopoietic pathologies such as cardiovascular disease. And so the incidence of clonal hematopoiesis, which is defined as a greater than 2% variant allele frequency of a particular clone with a cancer driver mutation in your blood, the incidence of this increases with age and over time, becoming particularly prevalent over the age of about 65. And if sequencing studies have shown that the mutational spectrum of genes which are recurrently mutated in individuals with clonal hematopoiesis is highly reminiscent of the mutational spectrum of our favorite age-related myeloid malignancies, such as AML, MDS, and myeloid proliferative neoplasms. And interestingly, for our lab research, the top 3 most recurrently mutated genes in CH all involve in epigenetic regulation of stem cells, DNMT3A, which is a DNA methyltransferase enzyme, TET2, which removes 5-methylcytosine marks from the genome, and ASXL1. So the genome sequencing revolution has now shown us that about half of all adult AML and MDS patients bear a disease that's driven by some genetic mutation in a component of the epigenetic machinery, something that modifies DNA methylation or histone modifications. Now, the functional importance of these mutations is underscored when we model them in genetically modified mice, showing that basically all of these mutations impact some important functional property of the hematopoietic stem cells, having impacts on their self-renewal, proliferation, differentiation, or apoptosis. Now, the gene that we've been particularly interested in studying over the last 9 or 10 years, as I mentioned, is a gene called DNMT3A, which encodes a DNA methyltransferase enzyme, which adds DNA methylation marks to the genome to regulate the epigenetic machinery. So I'm not going to go too much into the background about the molecular biology of DNMT3A, but just to suffice to say that it's the most recurrently mutated gene in CH, and also one of the most recurrently mutated gene in myeloid malignancies. But really, this is kind of the process that our lab is interested in studying. So we have normal hematopoiesis, which we're born with. Over time, these clones will gain mutations, which allow expansion and clonal dominance of a clone with a particular mutation, establishing this condition known as clonal hematopoiesis, a proportion of which patients will undergo further clonal evolution to myeloid neoplasia. And so really our lab is interested in understanding the cell intrinsic factors that these mutations corrupt to enable them to gain clonal dominance and outcompete their neighboring stem cells. We do that through a variety of low input genomic assays, as well as performing CRISPR screens, which I'll talk to in the second half of the talk, to understand genetic determinants of the increased resilience of these cells as a pathway to understanding targetable factors. But there's also a contribution from the cell extrinsic factors that change with age. So basically, we're interested in understanding how environmental factors change with age that might provide a selective pressure which favors the growth of mutant clones. And I like to think of this in terms of Darwinian evolution on a micro scale. So something changes in your environment, which then might provide these selective pressures, which favors the growth of clones with specific mutations that can tolerate this new stress. And again, there are multiple factors that we're trying to interface with the changing environment with specific mutations. And these can be broadly grouped into inflammation, diet, and the microbiome. For the purpose of today's talk, we're just going to talk about our work with inflammation. So inflammaging has been termed as a consequence of a cumulative lifetime of exposure to antigenic load. And chronic inflammation is detrimental to long-term stem cell function and typically leads to enforced differentiation and depletion of your stem cells over time and age. And repeated infections in people or patients with autoimmune disease have increased lifetime risk of development of AML and MDS. Again, implying that inflammation might be an important cell intrinsic factor, which selects for clonal evolution. So our hypothesis starting out this work was that inflammation imparts a selective pressure that confers a fitness advantage to clones with specific mutations. And I will say this was started 4 or 5 years ago, and this is now a popular idea in the field, so it's no longer unique to our lab. A lot of this work was started in parallel with my former colleague at Baylor College of Medicine, Dr. Catherine King, who works on microbial models of infectious stress. And Catherine and I had done an experiment where we created hematopoietic chimeras in mice mixed with wild type and DNMT3A mutant bone marrow, which Catherine then infected with a pathogen called Mycobacterium avium. And what you can see over here in this panel is that the mice that received the DNMT3A mutant bone marrow, which then underwent infection, had a massive expansion of their hematopoietic stem cell compartment compared to uninfected mice and control bone marrow. Here at WashU, we were in parallel performing a genomic study of an autoimmune disease cohort of patients with increased inflammation, which might provide kind of a natural model system of the effects of inflammation on hematopoietic stem cells. So we had performed genome sequencing on the blood of patients with ulcerative colitis. And what we found in the gray bar is that compared to a lot of the other population studies, our ulcerative colitis patients had elevated levels of clonal hematopoiesis, which was typically associated with increased mutations of DNMT3A, but also a gene called PPM1D, which I'm not going to talk about today. Catherine then went on to show with mouse studies with our help that this response of increased clonal expansion of DNMT3A mutant bone marrow under infectious stress required the intact interferon gamma receptor 1. When you knock it out, this response goes away. And we also went back to our ulcerative colitis patient cohort, and we got serum from these individuals and did an ELISA for interferon gamma. And we showed that when we stratified the patients into those that were clonal hematopoiesis CH negative, clonal hematopoiesis positive, but without DNMT3A mutations, and clonal hematopoiesis with DNMT3A mutations, that this last cohort had increased levels of serum interferon gamma. So these findings kind of formed the preliminary data which shaped our hypothesis, which was that somehow specifically interferon gamma acts as a selective pressure that favors the expansion of DNMT3A mutant clones in clonal hematopoiesis. And that somehow DNMT3A mutations might make hematopoietic stem cells resistant to the deleterious effects of chronic inflammatory signaling. And so the workhorse of our mechanistic studies are genetically modified mouse models. So we have 3 distinct models to recapitulate the spectrum of mutations found in DNMT3A and clonal hematopoiesis. Those are DNMT3A heterozygous loss of function and homozygous loss of function in the hematopoietic stem cell compartment, combined with a hotspot point mutation that's the most recurrent mutation found in AML. So we've generated these mice, and then we've also additionally crossed them to interferon gamma receptor 1 knockout animals to see the effect of removing type 2 interferon signaling on function of DNMT3A mutant stem cells. So one of the first experiments we did to test the functional potential of these stem cells was we took our control and DNMT3A mutant mice, and then we injected them with recombinant interferon gamma. And so one of the most well-described functional consequences of stimulation with interferon is that the stem cells become sensitive to apoptosis when they undergoing subsequent secondary stress. So what we did is inject the mice with recombinant interferon and then sort 500 stem cells into wells of 96 well plates. And then we assayed apoptosis by Cleese caspase 3,7 activity, either in freshly isolated stem cells or stem cells that were given a 16-hour secondary stress by overnight culture in minimal media. So this work was started by a graduate student in my lab, Beth Ostrander, now continued by a postdoc, Christine Zhang. So all the credit goes to them. So just to show you the results of this experiment, basically when we assay apoptosis in stem cells freshly isolated from mice, we don't see really any caspase activity at all. So stem cells are very resistant to stress in vivo, and this is consistent with many other studies we've seen in the past. But when we subject them to a secondary stress by overnight culture, what happens in control stem cells given PBS, which is our vehicle in this experiment, they show an increase in caspase activity compared to the unstressed cells, which is understandable. But really control stem cells which were treated with interferon gamma show a markedly increased apoptosis response compared to their PBS treated comparators. Now, what happens in DNMT3A mutant stem cells treated with interferon is that we don't see this increased apoptotic response. DNMT3A knockout stem cells treated with interferon in vivo are basically indistinguishable from those treated with PBS. So this told us that DNMT3A mutant stem cells were resistant to the deleterious effects of interferon gamma exposure. So we wanted to know if this was a specific effect to interferon or if this might be a conserved response of DNMT3A mutant stem cells to all kind of cytokines. And so when we did the same experiment with closely related cytokines such as interferon alpha, TNF alpha, or IL-6, we actually did not see any difference in cleave caspase activity from DNMT3A mutant stem cells exposed to these cytokines compared to control stem cells exposed to the same cytokines. So this is telling us that this is a relatively specific effect linking interferon gamma to DNMT3A mutations. And again, up front, I'll say that this is in no way specific. This is not the only mechanism that's going to promote clonal hematopoiesis. We just think this is quite a specific phenotype. And then when we extend the experiment across all of the DNMT3A mutation genotypes treated with interferon gamma, again, we see the same resistance to apoptosis in DNMT3A heterozygous and R878 stem cells treated with interferon gamma compared to control stem cells treated with interferon gamma. So it's specific and consistent phenotype. But we really want to know what happens chronically to stem cells treated in vivo with interferon over time. And so to understand this, we created hematopoietic chimeras by transplanting lethally irradiated recipient mice with a fixed amount of standard competitor whole bone marrow from wild-type mice in competition with the same number of whole bone marrow cells from one of the 4 genotypes listed below, control mice or those of our different flavors of DNMT3A mutation. Then, after stable hematopoietic engraftment, we treated mice in 2 separate treatment blocks with a variety of hematopoietic stresses. So we have PBS as our control, and then we have recombinant interferon gamma, PIPC and LPS, which are other stresses of the immune system. And we also included 5-FU, which is a chemotherapeutic that kills actively cycling bone marrow cells and forces quiescent hematopoietic stem cells into proliferative cycle to replenish the bone marrow system. And we're just going to cut to the chase and show what happens in the bone marrow at the end of the experiment. And really, we're interested in the long-term hematopoietic stem cells, phenotypically gated to look like this. So for any flow aficionados out there, this is the final gate of our flow cytometry plots showing phenotypic quantification of our hematopoietic stem cells. So at the top, we have control recipients treated with PBS. The gate shows the frequency of hematopoietic stem cells in our bone marrow over time. You can see below in this mice treated with transplanted with the same bone marrow treated with interferon gamma, we lose stem cells over time, which is a consistent result that's been reported in the literature. And then we also gate to markers of our donors and recipients. So CD45.1 is the internal control bone marrow. CD45.2 marks our various test cells. And you can see that treatment with interferon gamma causes depletion specifically of our control test cell population. So what happens to the DNMT3A mutant bone marrow over time? So those treated with PBS, you can see that we have this increase in stem cells in this phenotypic gate at the end of the transplant. Again, this is a consistent phenotype we've seen for the last decade. But now when we treat these mice with interferon gamma, we do not see a depletion of stem cells. And basically, interferon gamma doesn't touch the contribution of the donor-derived DNMT3A mutant stem cells to this cell fraction. So we can quantify the number of donor-derived stem cells per mouse at the end of the transplant as follows. And we can see that basically anything that causes replicative stress and proliferative stress of control stem cells leads to a loss of self-renewal over time. And this population is not able to sustain itself as well as PBS-treated animals. What happens for DNMT3A heterozygous stem cells, we see that they're similarly depleted by PIPC and 5-FU, less so by LPS, but interferon gamma doesn't touch the clonal expansion of these cells. We see a very similar result from DNMT3A homozygous loss of function. They're depleted by a lot of immune stresses, but not by interferon gamma, and the same result for DNMT3A R878. So this tells us, while in general, DNMT3A mutant stem cells are impacted in a similar way to normal stem cells by a lot of proliferative stresses, they are resistant to interferon gamma mediated stem cell depletion. So really the gold standard for functional quantification of stem cell activity in our field is bone marrow transplantation. And to understand the role of type 2 interferon signaling in the function of DNMT3A mutant stem cells, we crossed our DNMT3A conditional knockout mice to interferon gamma 1 receptor deficient mice. And so then what we do is we isolate 200 HSCs from the various genotypes and transplant them into lethally irradiated recipient mice, again, along with a fixed dose of internal control wild type bone marrow, so that we can really see in competition what is the effect of these mutations. And so we have several parameters that we measure through these transplants to assay the function of the stem cells. One is differentiation into peripheral blood. So we monitor the chimerism of our test cells to myeloid, B cell, and T cell fractions at 16 weeks post-transplant. So you can see the contribution of the control stem cells to the various cell compartments. Then when we take away interferon gamma receptor 1 from these control stem cells, we don't really see any impact at all in terms of the differentiation potential of these stem cells. When we transplant DNMT3A heterozygous stem cells, taking away interferon gamma receptor 1, again, really doesn't impact their differentiation in the peripheral blood. But more so when we have the more severe phenotypes of DNMT3A knockout and DNMT3A R878, what we see is that loss of interferon gamma receptor signaling does impact their differentiation into peripheral blood, particularly cells in the myeloid lineage. And this is important to us because really these mutations predispose to diseases of the myeloid lineage. So loss of interferon gamma signaling leads to modest effects in DNMT3A mutant stem cell differentiation, and as I mentioned, particularly in the myeloid lineage. But we really care about stem cells. And so we quantify, again, the number of stem cells which are derived from our test cells in the bone marrow of the mice at the end of the transplant. And again, what we can see is that loss of interferon gamma receptor 1 significantly inhibits the clonal expansion that we see from the DNMT3A knockout and R878 stem cell genotypes. This could be quantified either in terms of the overall frequency of these cells in the bone marrow or in terms of their absolute number. So whichever way you want to slice it, we can say that interferon gamma signaling is required for the clonal expansion of DNMT3A mutant stem cells. So this is really the first evidence that this could be a pathway that could be inhibited to prevent clonal hematopoiesis. Okay. So one could make the argument that, well, if you take away this signaling pathway, perhaps it just impacts the differentiation and self-renewal of all stem cells of any genotype. So to try to answer that question, we performed the same experiments, but this time with the second most frequently recurrently mutated gene in clonal hematopoiesis, that is TET2. So we generated TET2 conditional knockout stem cells and crossed them with interferon gamma 1 receptor knockout mice and performed the same competitive transplant experiment. And to summarize, taking away interferon gamma receptor 1 from TET2 knockout cells did absolutely nothing to inhibit their clonal advantage and their differentiation into myeloid, B cell, and T cell lineages. So you can see that there's no difference from TET2 interferon gamma receptor knockout myeloid differentiation compared to TET2 knockout alone, and that all of the TET2 genotypes have increased peripheral blood output compared to control. And importantly, when we look at the donor-derived stem cells in the bone marrow at the end of the experiment, again, we saw no impact in terms of stem cell numbers across any of the genotypes. So this at least gave us confidence that interferon gamma signaling is kind of not a universal clonal hematopoiesis selector. At least it had no impact for the impact of TET2 mutant stem cells, and that this effect might be specific to DNMT3A mutant stem cells. Okay, so that's mice. We had an inkling from our ulcerative colitis patient sequencing experiment that this signaling pathway is also important for human stem cells in vivo. But to more directly test that, we set up the following experiment. We took CD34 positive stem cells from cord blood specimens, and then we engineered DNMT3A mutations with CRISPR-Cas9 genome targeting. We then transplanted the targeted CD34 positive cells into NSG mice and basically recapitulated the experiment we did with mice. So we treated the recipients in 4-week treatment blocks, either with PBS vehicle or recombinant interferon gamma. And at the end, we assessed the effect in terms of blood cell chimerism and stem cell number. So this is a plot just showing the variant allele frequency of our targeted alleles in the CRISPR experiment. So orange represents the rate of modified alleles from our targeting. So we have two guides that we've tested to specifically ablate DNMT3A to create a mutant cell, and AAVS1 is our negative control, which targets an inert locus in human stem cells. So we get consistent targeting across all of our three genotypes. And so when we look at the peripheral blood contribution of human cells over time, we can see that comparing the different guides treated with PBS versus interferon gamma, we don't see a huge effect in terms of peripheral blood contribution. This is quite similar to what we saw with our mouse experiments. But really, again, we care about more so what happens with stem cells over time. So we use the following markers to now gate human stem cells by flow cytometry. And what happens when we do this is we can see that in PBS treated mice, we do see a bit of a clonal expansion of CD34 positive cells targeted with DNMT3A guide RNAs compared to the AAVS1 in PBS treated mice. But we show that this expansion is slightly elevated from treatment with interferon gamma, and whereas the control AAVS1 stem cells are depleted by interferon gamma, although not significantly because of the limited replicates, that this is significant when you compare to DNMT3A targeted cord blood cells. So again, these human cells are much more variable than mouse cells. We have ongoing replicates to increase the statistical significance. But really, the more important effect is doing a serial transplant, which is currently ongoing, by taking these stem cells into secondary mice to kind of mimic what happens with age in people. Okay, so hopefully the functional data has convinced you that this pathway is important for the clonal expansion of DNMT3 mutant stem cells. So the next step is understanding why. And so to begin to understand the molecular mechanisms of why this signaling might be important in particular stem cells of different genotypes, we performed an RNA-seq experiment on stem cells of control and DNMT3 mutant genotypes treated in vivo with either PBS or interferon gamma. And to summarize a lot of data and cut a long story short, we actually saw very few changes in gene expression across the genotypes treated with interferon gamma. And most of the changes that we saw were driven by treatment, so that the sample stratified by interferon gamma or PBS rather than genotype. So that was a lot of work for a relatively little payoff, although we did get some interesting findings out of it. So if we do gene set enrichment for pathways which are differential between control and DNMT3 knockout stem cells treated with interferon gamma, what we saw consistently was that different molecular pathways associated with cell cycle were not being engaged in DNMT3 mutant stem cells treated with interferon. So this kind of pointed to us that somehow cell cycle was defective in the mutant stem cells following treatment with interferon gamma, which normally causes proliferation of control stem cells. And just to stratify, just to pull out one of these gene ontology categories of cell division, we can see that basically all of the genes are enriched in the control stem cells treated with interferon gamma, and that these genes just aren't effectively turned on in DNMT3 knockout stem cells treated with interferon. So this kind of hinted to us that somehow the DNMT3 mutant stem cells were resistant to proliferation in the face of interferon gamma challenge. So this is something we can easily test in our lab. So we have mice of the different genotypes, as you can see, and then we treat them with a BRDU loading experiment. And this will basically tell us, in the presence of BRDU, how many stem cells have divided over the course of a particular treatment period. So the number of BRDU positive stem cells over the different time course. And then we treat mice during this time course with either control PBS or interferon gamma and read out the effect by flow cytometry. And so this is kind of the effect, and I'll walk you through the results here. We're looking at the percentage of BRDU positive stem cells at the end of a 72-hour treatment block. So what we can see is that DNMT3 mutant stem cells are more quiescent than control stem cells under homeostatic conditions. They have less BRDU positive stem cells over this treatment block, and this is consistent with results we've seen in the past. But more importantly, DNMT3 mutant stem cells are less activated in response to interferon gamma than wild-type stem cells. So wild-type stem cells treated in vivo with interferon show a significant increase in proliferation, a response which is lacking in the DNMT3 mutant genotype. And again, we wanted to show that this was just not to an overall inability for DNMT3 mutant stem cells to escape quiescence. So we also treated mice with separate mice with 5FU during this treatment block. And basically, stem cells of all genotypes responded similarly to 5FU and go into cycle. So this was the first evidence that we had that the inability of the DNMT3 mutant stem cells to engage cell cycle might underlie their functional properties. And to really test this on a clonal level, we performed the following experiment, which looks complicated, but I'll try and break it down for you. So we wanted to assay the response of individual stem cells to interferon gamma. And so what we did was to cross our control DNMT3 mutant mice with mice that had doxycycline inducible labeling of H2B GFP. So what happens in these mice is you could give them doxycycline chow, which will then load histone 2B GFP into the nucleus. And we use this basically as a way to track stem cell division. So after the labeling period, we then perform basically the same BRDU experiment. We treat mice with PBS or interferon, but this time we sought single GFP positive HSCs into individual wells of 96 well plates and count the time to first stem cell division at 12-hour intervals through the microscope. And this is the result that we get. So control mice treated with PBS show the following dashed line, and basically an important metric here is the time to the first division, the average time to first division of the single stem cells. And when we treat them with interferon, what happens is this shortens the time of control stem cells to their first division. So this is good. Now, when we compare DNMT3 mutant stem cells treated with PBS, we really see no difference in time to first division compared to control stem cells at baseline in mice treated with PBS. But what happens is when we treat these mice with interferon, the time to first division of these stem cells does not change compared to treatment with PBS. So this is telling us that, again, this is another way of showing that DNMT3 mutant stem cells are resistant to going into cell cycle following exposure to interferon. And another thing we noticed from this experiment was even at the end of the 72-hour counting period, a time point when virtually all of the control stem cells had proliferated, there was a population of DNMT3 mutant stem cells, about 30% of their population never proliferated at all. They just sat there looking at you for the entire treatment block. So again, this pointed to some level of clonal heterogeneity of this response, even within our highly immunophenotypically purified cell population. And so to understand if there might be specific subtypes of stem cells that respond differently to interferon gamma in the bone marrow, this led us to perform a single cell RNA-seq experiment on the 10x genomics platform. And I think by now, most of you are probably familiar with the workflow of single cell RNA sequencing. So I'm not going to go into the technical details, but I'll just cut to the chase of our findings. And basically, again, to cut a long story short, we found that either control or DNMT3 mutant stem cells treated with interferon in vivo, then sorted for single cell RNA sequencing, we could not actually stratify the genotypes by treatment, and that we found that when we clustered all of the cells together, what happens was that cells were clustering by treatment, so into PBS or interferon rather than genotype. So the treatment effect is stronger than the genotype effect, which was the same as what we saw from our bulk RNA sequencing experiment. But we were able to do some nifty tricks using the single cell RNA-seq. So if we just plot all the cells from our experiment on a UMAP and then assign cell identities using the expression of specific marker genes within the clusters and cell populations, we are able to resolve specific clusters of cells within our experiment, which represent different progenitor cell populations in the bone marrow. And then when we do this and we analyze the differentially expressed genes in these specific populations, we were able to pull out about 50 differentially expressed genes in the different stem cells across the different genotypes. And so we sort of scoured this list for the expression of differential genes, which might kind of help explain the phenotype that we were seeing. And one gene which caught our eye was a gene called Txnip, which is about twofold upregulated in the DNMT3 mutant stem cells at baseline and further increased in expression after interferon gamma, an effect that was not seen in the control stem cells. And this gene became interesting to us because of its, there were reports in the literature about it has important functions in stem cells under stress. And we were particularly interested in a potential interaction between this gene, which normally acts in metabolic pathways, but we're interested in this potential interaction with P53. So this is where Txnip is typically expressed in our single cell RNA-seq data. So it's largely expressed, but not specifically expressed in the stem cells. And then we also quantified, performed a follow-up experiment in parallel to confirm the experiment by isolated real-time PCR. So we see that in DNMT3A mutant cells treated with PBS, there is an increased expression of Txnip compared to PBS-treated control stem cells. And while the mutant stem cells don't change in expression of Txnip after interferon, the control stem cells go down. We also validated that this happens at the level of protein by Western blot. So Txnip was increased in the DNMT3A knockout mice treated with interferon gamma by Western. So hopefully it's a conserve between RNA and protein. And as I mentioned, we became interested in this gene because of its potential interaction with Txnip. So we've also not going to be presented here today for the sake of time. We've done DNA methylation profiling in progenitor cells exposed to interferon chronically. And so we're able to correlate some DNA methylation differences also with our gene expression. So kind of to summarize the model when we put out RNA-seq and methylation data together is the following. We think that under normal circumstances, DNMT3A targets the Txnip methylation regulated enhancer element, and this kind of regulates its expression under normal conditions. Now, under these normal conditions, P53 is targeted by a protein called MDM2 for post-translational ubiquitination. And this ubiquitination leads to proteasomal degradation of P53 in the cell. Okay, what happens when you have a mutation in DNMT3A is that this regulatory element of Txnip becomes hypomethylated, which increases expression of Txnip in DNMT3 mutant stem cells. Now, our hypothesis from the literature was then that this would then free up Txnip to interrupt this interaction between MDM2 and P53, which would then lead to stabilization of P53 protein and allowing it to then go through a number of target gene expression and elicit functional consequences. So in line with this, we don't see any changes in P53 transcript levels across our stem cells under different treatment conditions. However, we do see a stabilization of P53 at the protein level in DNMT3A mutant animals, both at baseline and after treatment with interferon gamma. So this led us to the hypothesis that this P53 stabilization might be important for the functional resistance of DNMT3 mutant stem cells, although we did not see a global signature of P53 target genes in the RNA-seq. And this may be because of the diverse functions of P53 in the cell in response to inflammation. So P53 has diverse functions in terms of survival, apoptosis, and cell cycle. So to understand, if there might be specific categories of genes which are influenced by P53 in our mutant stem cells, we kind of picked a few genes for assay of gene expression in different categories. So for survival, some of the important P53 target genes include BCL2 and MCL1. We don't see any difference in expression there. In terms of pro-apoptosis, important P53 target genes are BAX and PUMA, and we don't see any changes there. But really the big winner that we saw was the classical P53 target gene, P21. So P21 was increased in our DNMT3 mutant stem cells at baseline and then further augmented in response to interferon gamma. And I've got to say, after about 9 or 10 years of doing these types of experiments, this was one of the biggest changes we've ever seen. So P21 is a cell cycle inhibitor, and kind of this led us to the following overall refinement of our model, in that in the absence of DNMT3A, Txnip stabilizes P53, which is then targeted to CDKN1A, which leads to increased expression of P21. And this is a cell cycle inhibitor, which would then increase the expression of this cell cycle inhibitor, would enforce stem cell quiescence after treatment with interferon, which then would retain their functional potential, as the preservation of quiescence is one of the key features of stem cells with long-term functional potential. And really kind of to put the nail in the coffin for this story, We're doing genetic rescue experiments where we do shRNA knockdown of Txnip, P53, and P21 in control and DNMT3 mutant stem cells to see if it normalizes the response of these cells in response to interferon gamma. And our functional readouts are apoptosis, cell division, and in vivo by transplantation using all of the experiments that we've talked about already today. This is just showing you that we've designed effective shRNAs. These experiments are already underway in mice. We've got the first cohort, and it actually does validate our hypothesis. So hopefully this will be kind of the end to this first part of the story. So I'm going to summarize the first part of the talk as follows. So what we've concluded thus far is that DNMT3 mutant stem cells are resistant to the deleterious effects of interferon gamma, that interferon gamma signaling is required for the clonal expansion of DNMT3 mutant stem cells in vivo. The DNMT3 mutant stem cells retain quiescence when challenged with interferon gamma. And we think this Txnip P53 CDKN1A axis sustains DNMT3 mutant stem cells under chronic inflammatory stress. So our ongoing studies include genetic rescue experiments to validate our pathway and our hypothesis. We're completing the parallel experiments with primary human stem cells in NSG xenografts. And the next question is kind of, is this signaling relevant to the malignant progression and clonal expansion of DNMT3 mutant stem cells, which is kind of a separate question. And really, so if we prove that this is, hopefully I've convinced you that this is important, at least in model-based systems for the development of clonal hematopoiesis. And so if this is important, then this raises the question to us is what we're really interested is in finding ways to prevent clonal hematopoiesis and malignant progression in elderly people. And so this is just a representative schematic of what happens in the clonal evolution of MDS. So we have these pre-malignant stem cells which establish clonal hematopoiesis. Eventually they have a second bad day and they run into a relevant cooperating mutation that drives progression of clonal hematopoiesis to MDS. So for MDS, we currently have 3 FDA-approved therapies, none of which are curative. So the only cure is bone marrow transplantation, but a lot of these patients are ineligible due to age and the associated comorbidities. So really what we're trying to do with our lab studies is develop methods which would selectively inhibit or eliminate these emerging pre-leukemic stem cells during this clonal hematopoiesis phase. And we want to actually prevent people from developing MDS or AML rather than cure patients with the mature disease. So this would be a statistical reduction of the probability of an at-risk individual acquiring the relevant secondary mutations necessary for clonal progression. So given what I've shown you in the preceding slides, could reduction of interferon gamma signaling represent a viable method for reducing DNMT3 mutant clonal hematopoiesis as a precursor for inhibition of blood cancer development? And so kind of the first lab-based experiment to test this is to simply say, what if we take control in DNMT3 mutant stem cells, and now we transplant them into an environment of interferon gamma deficiency? So now we perform our competitive transplant experiments, but this time instead of transplanting into wild-type mice, we transplant into mice which are interferon gamma deficient, so they can't make any interferon gamma. And basically what happens if we look at the blood for our engrafted cells, so we have wild-type stem cells engrafted into either wild-type or interferon gamma knockout recipients, and we have DNMT3 knockout stem cells engrafted into wild-type or interferon gamma knockout recipients. What we see is that transplanting either genotype into an interferon gamma deficient environment hurts their peripheral blood differentiation, but it's similar manifested by both genotypes. But more importantly for us, what happens to stem cells in the bone marrow at the end of the experiment? So DNMT3 knockout stem cells transplanted into a wild-type environment, we see this expansion of this population, this clonal expansion, compared to wild-type cells going into wild-type mice. So wild-type stem cells going into interferon gamma knockout mice are not inhibited, show no difference in their numbers at all. But now DNMT3 knockout stem cells going into an interferon gamma deficient mouse, we do see a reduced clonal expansion of this population, telling us that environmental interferon gamma is required also for the clonal expansion of DNMT3 mutant stem cells. So with that finding in hand, we then turn to the question of what is the translational potential of this finding? And so we wanted to know if there's a pharmacological equivalent we could use to test this hypothesis. So reading the literature, we found this recent report that the bromodomain small molecule inhibitor JQ1 is an agent that could be used to reversibly block interferon gamma production. So using this, we use JQ1 to pharmacologically inhibit interferon gamma production in our competitive transplant model. So again, we have competitive transplantation of control or DNMT3A knockout stem cells, this time all into wild type mice. And then mice were randomized to receive vehicle-controlled DMSO or treatment with JQ1. And just to cut to the chase and show you the result we really care about is what happens to stem cells in the bone marrow. Again, we see a clonal expansion in DNMT3 and knockout stem cells treated with vehicle compared to the vehicle-treated control stem cells. Control stem cells treated with JQ1 are not inhibited, but DNMT3 knockout stem cells treated with JQ1 are significantly impeded in terms of their degree of clonal expansion. So pharmacological inhibition of interferon gamma is also able to reduce clonal expansion of DNMT3 mutant stem cells. So we really think that this could be a viable way to reduce clonal hematopoiesis and the burden of DNMT3 mutant clones in the bone marrow of individuals with clonal hematopoiesis as a mechanism of blood cancer prevention. So this experiment just used JQ1 as a proof of concept. There are now more specific bromodomain inhibitors, which we're working with GSK to try and test in our model to see if we can get an even better response. And also this led us to the question of what is kind of the genetic basis for the susceptibility of the DNMT3 mutant stem cells to JQ1. And this is where the Longer Life Foundation funding comes into play. So we have been doing CRISPR-Cas9 negative selection experiments to try and understand genetic determinants of the molecular phenotypes of DNMT3A mutant stem cells. So one of the phenotypes of this clonal expansion and increased self-renewal is that we can serially replate these stem cells in methicult media. Again, not to belabor the details here, but just to get to the important point, is that after about 4 passages, we can grow these cells as liquid cells, as liquid cultures, not indefinitely, but long enough that they give us cell lines with the epigenetic identity of primary stem cells, which are amenable to biochemical and genetic manipulation. And so the tool that we used for these experiments are CRISPR-Cas9 negative selection screens. And the goal of these experiments are to identify in an unbiased fashion what are genetic or epigenetic dependencies that are selectively required for the growth of DNMT3 mutant stem cells, but not normal wild type stem cells. So this is to find targetable genetic factors to slow the clonal expansion of DNMT3 mutant stem cells in individuals with clonal hematopoiesis. And specifically the work that was funded by the Longer Life Foundation was that we took individual stem cells from our genetically modified mice that express Cas9, and then we expand them and then transduce them with guide RNA libraries that express GFP. And so this is all in targeted, not genome-wide, but we have various focus screens that we're doing. We have 4 to 5 guide RNAs that target individual genes in our lentivirus pools. We transduce the cells, and then we just simply sample the DNA in the transduced cells at different time points throughout a 3-week culture period. Because the guide RNAs have Cas9, we can do targeted experiments by flow cytometry, or we can do pooled experiments by next generation sequencing to get our answers. And really what we're looking for are dropouts, what we call negative selections. So we're trying to identify guide RNAs that drop out of the lentivirus pool over the experiment. So this would indicate that what the particular cell that received that guide RNA was killed or outcompeted over the culture period, saying that this guide RNA targeted a gene which was crucial for the growth of that cell. And so these are genes that we're interested in targeting. Okay, so just to summarize kind of one particular element of the next generation sequencing bioinformatic readout, we identified genes which are targeted by multiple guide RNAs which become depleted from our pool over time. So again, we highlight them through a number of different bioinformatic filters to find out kind of candidate genes that we wish to pursue forward into functional assays. So once we had our refined list of target genes, we do a follow-up targeted experiment in vivo or using primary cells. And so we take mice that express Cas9 cross to our DNMT3A conditional knockout alleles. So we take, they also express GFP. So we take the stem cells, transduce them this time with individual guide RNAs targeting our genes of interest. And then we sort stem cells and do a replating assay in vitro to see the effect of the specific guide RNAs on the growth of our stem cells. And so what we're looking for as a functional readout of these experiments is we're looking for guide RNAs that target genes which reduce the colony-forming capacity of specifically of DNMT3A mutant stem cells, which do not inhibit the colony-forming capacity of our control stem cells. And so you can see from this particular figure, we've identified a few genes which we kind of selectively target our DNMT3A mutant stem cells and inhibit their function without significantly inhibiting function of control stem cells. And for the sake of time, we're just going to show the follow-up functional experiments with one, which is a gene called BRD2. And you'll see why I'm going to show you that in a minute. This really was the beautiful dovetailing of a couple of different projects in our lab. And so we thought BRD2 was a very interesting gene to follow, to move forward into our functional experiments. And so that the previous slide was our in vitro validation, and now we move forward into in vivo validation. So to do this, we target Cas9 expressing control stem cells or DNMT3A mutant stem cells with our non-targeting negative control guide RNA or guide RNAs targeting BRD2. So we transduce them with lentivirus expressing the guide RNAs, transplant them into lethally irradiated mice, and read out the effects in terms of blood and stem cell enumeration, as I've previously shown. So now we're just looking at the blood engraftment of control stem cells targeted with either the negative control guide RNA or BRD2 guide RNA. And you can see there's basically no effect in control stem cells by targeting BRD2. This was also reminiscent of our previous colony forming experiment. But now when we see DNMT3A knockout stem cells targeted with a non-targeting guide RNA in dark blue up the top, which have elevated peripheral blood chimerism. When we target BRD2 in the same cells, we have reduced peripheral blood chimerism, so it does inhibit their function. But more importantly, when we look at the bone marrow stem cells at the end of the experiment, we are now, again, ablating this clonal expansion of the DNMT3A mutant stem cells, which happens from the non-targeting guide. When we target BRD2, we're able to reduce the number of stem cells in the bone marrow. So again, we think that BRD2 inhibition could be a mechanism to slow the clonal expansion of DNMT3A mutant stem cells in vivo as a mechanism of blood cancer prevention. But why have I told you these two kind of parallel stories? Hopefully this one slide will summarize why I told you that story. And so we show that JQ1 treatment with the bromodomain inhibitor JQ1 was able to reversibly inhibit the clonal expansion of DNMT3A mutant stem cells in vivo. And so to understand the genetic basis of this, why is JQ1 selective for this? JQ1 is a bromodomain inhibitor, and the main targets of this are BRD2, 3, and 4. And so BRD2 was our main genetic factor that we identified from our CRISPR screen. And so we think that this really provides a beautiful mechanistic link going all the way from genetics through pharmacological inhibition to translational studies. And so this really provides, in our mind, good evidence that this pathway is important in clonal hematopoiesis for these mutations under inflammatory stress, and that pharmacological inhibition of BRD2 via JQ1 or more specific small molecule inhibitors that we're developing represents a very attractive potential pharmacological means to reduce clonal hematopoiesis in individuals, because JQ1 is very well tolerated in people. It's already in clinical trials for a variety of other solid tumor malignancies. And the pharmacodynamics are very good. It's non-toxic. It's very well tolerated. So we think that this could be a very attractive pathway to inhibit in otherwise healthy individuals with clonal hematopoiesis as a mechanism of blood cancer prevention. And so just to kind of come back to the model and summarize really what we're trying to do here. So we're interested in understanding this clonal evolution as we go from normal hematopoiesis to clonal hematopoiesis and myeloid neoplasia. And although over the last 5 to 10 years, we really have a very complete and detailed understanding of kind of the genomic trajectory of myeloid neoplasia and aging, this is not really translated yet into clinical benefit. Although we're developing now different targeted therapies based on specific mutations, this has not translated for the patients in terms of overall survival benefits. You know, everybody's trying now venetoclax with azacitidine in their favorite malignancy. We have specific IDH2 inhibitors, which is a very specific targeted therapy for individuals with IDH mutations. And FLT3 inhibitors, I don't think are going to prove effective for various reasons, but they don't target the founding clone in these malignancies. So although genetics has really given us a good understanding of the biology, what's lacking is a translation of this understanding into clinical benefit for these patients. And really the whole concept of what our lab is trying to do is to try to say, like, if we can't, if treating the mature patient is too late, what if we could actually stop these patients from getting to blood cancer in the first place? And so we're trying to work on what I call precision intervention to identify individuals with high-risk clonal hematopoiesis and actually try and treat them up front and stop them from progressing to blood cancer. So we're interested in developing and translating our lab findings into mutation-specific, non-cytotoxic targeted agents. So we think that this could be an effective strategy for the prevention of AML, MDS, MPN, and potentially a range of other blood disorders. And again, this might not be limited to diseases of the blood system, as clonal hematopoiesis is also associated with increased risk of cardiovascular disease, diabetes, and potentially some neurodegenerative conditions such as Alzheimer's. So such a strategy of precision intervention might actually have a substantial benefit for a wide range of patients. So with that, I'll kind of summarize and leave it at that. I'd like to thank everyone from my lab who's contributed to these studies over the last 4 or 5 years. Our key collaborators at Washington University, you can see is listed, and in particular, our funding bodies that especially the support from the Longer Life Foundation in getting some of the pilot studies with our CRISPR experiments off the ground, which would be unlikely to be funded by traditional agencies such as the NIH. So with that, I'll close and I'm happy to take any questions. Thank you.
Thank you so much, Dr. Challen, for taking us through all the work of your lab through the last years and the funding with the Longer Life Foundation being able to contribute to a lot of this work. We do have a couple of questions in the next couple of minutes that we can get to. The first one asks, so how would you determine who would receive BD1 and or BD2 inhibitors or the like in a general population? Would this be over the course of a lifetime, or when would one begin treatment?
This is the million, I would say this is actually the billion-dollar question in our field right now. Everyone is interested in understanding who is high-risk clonal hematopoiesis, how do we identify these people, and then what would you actually do about it? So for the first part, I think we're actually going to have a very good understanding of the patients who are the most at risk from epidemiological sequencing studies such as the UK Biobank and TopMed over the next 5 years. We're going to have a very good understanding with individuals with the highest likelihood to progress. Now, the second part is the hard part. What do you do about it? And those are the questions we're trying to answer with our lab studies. And even if we show we have a very specific effect for our JQ1 for people with DNMT3A mutant clonal hematopoiesis, the question then becomes, what kind of dosing strategy would you use? And for that, we really don't know. Do you need to give it to them chronically? Kind of like, do they need to be on like low dose aspirin, like that kind of thing for the rest of their life? But when you think about it, you don't really want to do that because you don't want to decrease old people's immune responses to fight infections. Or could you give it for like kind of higher dose for 2 to 4 week chunks of time to knock down that clone kind of below the level of detection again, and then you kind of repeat that every year or 2 years, and that's enough for cancer prevention of that clone. So these are questions that all need to be worked out, but I think it's a very exciting area to work in right now because I think this is really the way of the future.
Yeah, indeed. Okay, I think we have time for one more question here. So we have the question of how does your model fit in with the tumor immunotherapy models where interferon gamma and immunogenic signaling may be important for surveilling and eliminating malignancy with the immune system? He asks, are we doomed with both sides of the coin, and are JQ1 studies showing any increased incidence of solid tumors occurring?
Okay, so I'll start with the second part of that question because it's easier. JQ1 has proven to be remarkably ineffective in clinical trials against solid tumors. And I think probably because they're at a stage that's too late. The cancers by that point, they don't care about JQ1. I think if we can actually start treating cells in the pre-malignant stage, we have a much higher chance of success for bromodomain inhibitors. The first part is actually, yeah, it's a little bit daunting, and there's been work here that's shown treatment that inflammation might increase the immunogenicity of certain liquid tumors and solid tumors for cancer immunotherapy. And yeah, we have evidence in the lab that certain mutations might actually increase the interferon gamma output of specific clones. So I think the immunotherapy question is certainly a challenge at this stage, and I'd say it's very incomplete understanding.
Oh, thank you. And then last question here, would you expect that cytokine release syndrome in some stem cell transplant patients could potentially select for specific hematopoietic stem cells?
Yeah, absolutely. And that's already been shown in some studies. There are studies emerging now about tracking the genetics of the donor cells in BMT and then what happens post-transplant. And people are showing that you do get a clonal expansion of the healthy, normal stem cells in the recipients, and that this can be exacerbated by certain conditioning regimens or different kind of follow-up treatments. So the development of donor-related clonal hematopoiesis in bone marrow transplant recipients is actually now almost at the point where it's being a clinical consideration for these patients. And actually a recent study from a friend of mine, Coleman Lindsay at Dana-Farber, showed that DNMT3A mutant clonal hematopoiesis is actually beneficial for transplant recipients, and they actually do better. So again, it's a very complicated story. There's a lot to work out, but I think this is a really exciting area of new research.
Indeed. Thank you so much, Dr. Challen, for taking us through your story here and sharing with us all the work with your lab. I think that concludes questions today. If anyone has further questions, you can feel free to reach out to me or to Dr. Challen himself. Thank you for joining us this morning.